Um, Good afternoon, everyone, and welcome to our first Infogest Viva webinars. I'm Gaetan. This event was organized by a team of postdocs in the Food Research Center at Moore Park, Chagask. I will be starting the first webinar, and we would like to introduce you Talita Comunian, Shannon Gwala, and Daniela Freitas, unfortunately unable to attend today. We will be leading the next webinars every first Wednesday of each month. We hope you will enjoy the event and make the most of this opportunity. The goal of this webinar is to provide you the most recent findings in the field of digestions, coming from the PhD Viva defended over the last year, and of course to maintain the link in between the Infogest community. The webinar is right now broadcasted live on YouTube and will be accessible in the Infogest YouTube channel. The recorded presentation of today will be followed by a question-answer session with the speakers. So I'm inviting all of you to use the Q&A button at the bottom of your screen. Now I'm going to introduce our speaker, Yihong Chen. Yihong successfully defended his PhD in September in the Food Chemistry and Technology Department in the Chagask Food Research Center and in the Department of Biological Sciences at Munster Technological University here in Ireland. His project crossed areas including development of infant formula, lab and pilot scale unit operations, membrane filtration, thermal processing, homogenization, spray drying, but also worked on protein characterization and functionality, as well as in vitro and in vivo digestion. So right now, Yihong, the floor is yours.
Hello, everyone. My name is Yihong. Sorry. Hello, everyone. My name is Yihong Chen. I'm from Chagask, and my PhD project was using membrane-filtrated infant formula, the system to make the infant formula and assess the bioavailability and the bioactivity of the new infant formula. So I'm glad to be the first speaker of the Infogest to be the Viva seminar. So I hope you guys enjoy my PowerPoints and get some information from PowerPoints. Thank you very much.
Everyone, my name is Yihong Chen. I'm from Chagask and Munster Technological University in Ireland. It is my pleasure to become first speaker for the Infogest Viva webinar. My project was using a membrane filtration system to increase the native whey protein level in the infant formula, and I also tested its effects on the bioaccessibility and bioactivity. Currently, I have 3 publications in membrane filtration system and digestion of my new infant formula, and just 2 manuscripts have been submitted. The objective of my project is to humanize protein in infant formula closely resemble the breast milk. One of the most important differences between breast milk and infant formula is the breast milk is native. It is customized to the human infant and hence it's easier digested. In contrast, infant formula have to undergo several heat treatment processes, which lead to denatured protein ingredients. This will cause longer gastric emptying, altered peptide profile, and higher energy consumption. My project went through 3 sections. First is processing. Second is assess bioaccessibility by the in vitro digestion and in vivo digestion. The third section is assess bioactivity of my infant formula in cell culture and piglet growth. Let me introduce the first section, processing the infant formula by introducing membrane filtration. I mix the skim milk and whey protein, feed this preparation to a 1.4 micrometer microfiltration. At this stage, over 90% pathogens were retained in the retentate. The clean permeate further feeds into 0.2 microfiltration for filter the risk bacteria. The permeate were considering a member sterilized stream, and the retentate contained risk bacteria were mixed with lactose, fat, and minerals, then underwent heat treatment. Two sterilized streams were mixed together and fed to homogenizer, evaporator, finally spray dried to powder. I made my infant formula in 2 scales and analyzed the protein profile. The lab scale processing preserved only 18% native whey protein in the final powder. It is because the high contribution of retentate volume and dead volume to the feed volume during member filtration in such small scale. When I went to pilot scale, the level of native whey protein increased to the 60%. Also, I made the control infant formula using conservative heat treatment processing. The heat-treated infant formula contains significantly lower level of native whey protein. In the second section, to test the bioaccessibility of my infant formula, I firstly developed a same-dynamic in vitro digestion with infant pyramids. The pyramids of this model were referred in several literatures, and I list these pyramids here. The pH in simulated stomach will rise from 2.5 to 7.5 after infant formulas were ingested. There are videos during digestion and emptied digester assessed by CLMS. The green is protein and the red shows fat droplets. At the ingestion point and the first gastric emptying point, member-filtrated infant formula showed significant higher pH compared to heat-treated infant formula. This indicated to member-filtrated infant formula has a better buffer capacity. After 1 hour digestion, pH in gastric models declined to about 2. Some curds and protein aggregations were formed. At GE3, we can observe the member-filtrated infant formula form smaller curds and protein aggregations compared to the heat-treated infant formula. And an interesting observation here is the member-filtrated infant formula showed a phase separation in the vessel, a gelling curd phase and a stern phase, while the heat-treated infant formula had limited phase separation. At GE4, most proteins surround fat droplets were degraded, but heat-treated infant formula still has a lot of protein aggregations. If you look at the heat-treated infant formula vessel, you can see there's large curds. At the last gastric emptying point, fat droplets has collapsed in the member-filtrated infant formula, but the heat treatment infant formula still contained protein aggregates. Afterwards, I used the protein analytical method to analyze the protein degradation and peptide release. Member-filtrated infant formula contain higher water-soluble proteins and higher concentration of free amino groups in most gastric points. Furthermore, in member-filtrated infant formula, the water-soluble proteins will keep declining while they were stable in heat-treated infant formula at last 3 gastric points. Peptides were released reached at the peak at the gastric point 4 in member-filtrated infant formula. In contrast, heat-treated infant formula keeps releasing peptides until to the last gastric points. These observations showed the gastric coagulation kinetics were affected by processing and ingredients interactions. Also, I'm going to explain why curds flow up and how it affects the digestion. When a milk product underwent homogenization, proteins are embedded on the surface of fat droplets. Protein has a higher density than water, but fat has a lower density. Therefore, the protein leads curd segmentation, while the fat leads curds floating up. Compared to milk, infant formula contains less proteins. Also, in low native whey protein infant formula, more casein and denatured whey protein form the protein aggregations. They are easily embedded onto the fat due to their hydrophobic capacity. The member-filtrated infant formula with high level of native whey protein formed less protein aggregates and reduced emulsifiability. Here I established a hypothesis for different digestion performance between two infant formulas. The member-filtrated infant formula forms smaller protein and fat network due to the less protein on the surface of fat droplets, which has low density leads curds float up, and also they form the smaller protein aggregations. In the heat-treated infant formula, it forms larger fat protein network and larger protein coagulations, hence the curds were precipitates. Because the smaller networks and coagulations in the member-filtrated infant formula, the facilitated accessibility to the enzymes which increase the protein digestion. In contrast, large particles in heat-treated infant formula reduce the digestibility. The next stage, we made a small intestinal digestion and absorbed the digester using an in vitro cell model, the CACO2 monolayer. If you don't familiar with this model, you can simply imagine capital phase is intestinal lumen and bacillatory phase is the blood vessel. So what we observed here is there are more peptide and free amino acids cross the intestinal barrier in member-filtrated infant formula compared to the heat-treated infant formula. The next stage in this section is we feed our infant formula to the piglet. I listed the parameters here for the piglet trail. You can take a look. Using the analytical methods I developed during in vitro trial, we analyzed the protein degradation and peptide release in the piglet's digester. Similar to results I found in the in vitro digestion, I observed the member-filtrated infant formula released more water-soluble whey proteins and peptides. Also, releasing of free amino acid group were peak earlier than heat-treated infant formula. From release of individual amino acids, we can see the same trend. Member-filtrated infant formula released more amino acids in the jejunum compared to the heat-treated infant formula. Two important indicative amino acids I listed here. First is the viability of trypsin can be reduced by the heat treatment. We can see here the member-filtrated infant formula had a significant higher concentration of trypsin compared to the heat-treated infant formula. And the second is proline indicate to the peptides are easier to assess the amino peptides. This is also high in member-filtrated infant formula. Also, we can see here, in member-filtrated infant formula, amino acid releasing reach peak at the jejunum, but in the heat-treated infant formula, it's keep increasing until in the ileum. In the next section, let's see how the infant formula processing affects the bioactivity. The piglet and food intake data showed quite interesting results. So during the last 7 days of 28 days infant formula dietary intervention, the member-filtrated infant formula feeded piglets tended to have a lower daily weight gain and food intake. Also, member-filtrated infant formula results a lower total trace digestibility of crude protein. So if we extend our piglet trial, would a significant difference occur? To further assess the bioactivity of infant formula, I established a cell culture model. I picked 3 peptides from bacillary phase from each infant formula digestion. And also from the piglet trial, I collected the blood serum samples 3 hours after their first dinner at day 0 and 3 hours after their last dinner at day 28. Use these peptides and serums to grow the muscle cell and fat cells. The main results are the serum and peptides from member-filtrated infant formula increased the fat cell proliferation and adipogenesis compared to the heat treatment infant formula. So the above slides are all my 4 chapters result. Bring everything together, give a summary. The result is quite interesting. So the member filtration system to make the infant formula, it does increase the native whey protein in the infant formula, and it increase the protein digestibility, increase the peptide amino acid release, and also altered digestion kinetics. It reduces the crude size, form the flowed crude, and the serum from the piglet who feeds the member-filtrated infant formula had a higher a higher fat cell proliferation, and also the peptides increased the adipogenesis. However, from the piglet trial, we found the nutritional bioactivity, which is the body weight gain, is potentially reduced if we extend our trial, and also the crude protein of total digestibility is significantly low in member-filtrated infant formula. It is a very interesting result. I would like to give a hypothesis explanation to this scenario. Previously, a Canadian group feed piglets by 2 types of isocaloric modeled infant formula for 18 days. The difference is the protein profile. One diet is 100% whey protein and another is 60% whey protein and 40% casein. They found the whey protein diet group had a significantly lower body weight gain and food intake compared to the 6 to 40 whey protein casein diet. They explain that the whey proteins have a high protein quality. This increased the gastric emptying, satiating effect, and digestibility. Combining our results, I made a hypothetical explanation. So for the high native whey protein, the member-filtrated infant formula, after infant formula ingested, the infant formula coagulates due to the acidic and enzymatic coagulation. The casein, denatured whey protein, and fat, they formed a low-density network displayed as a floating crude phase, and the water phase contains water-soluble proteins, which mostly native whey protein. At the next stage, the water phase was transmitted to the small intestinal to digesting these whey protein first. Then the high quality native whey protein increase the satiety and appetite hormone lead to a thing called intestinal break. As a result of their intestinal break, brain tells the stomach to reduce the gastric acid secretion and gastric emptying, and tell the small intestine to reduce the enzyme and bile acid secretion. As a result, this reduces the digestibility of following gelled crudes. In another hand, the low native whey protein heat-treated infant formula after the ingestion. The ingredients form crude with no phase separation, and sometimes we can see the large particles. These crudes continually empty into small intestine, and the satiety and peptide hormones are maintained at the maintained level. Therefore, the intestinal breaker would be delayed or weakened. So this results gastric acids and emptying are maintained, and also the enzyme and bile acid. Therefore, the digestibility of these crudes are finally increased. So the difference, if we back to our results, the different peak of protein degradation can support this hypothesis actually. So the final question is, the new high native whey protein infant formula is good or bad? It altered digestion and it reduced the body weight gain compared to the heat treatment infant formula. The one problem of the traditional infant formula is related to a high energy consumption, and it leads to the overweight risk in the later life. So our further study will be compare native whey protein infant formula to the breast milk. Some other results we found from our infant formula is the low heat treatment can preserve the endogenous enzyme. One of indicator is archaein phosphates. Our member filtrated infant formula has over 28,000 archaein phosphates enzyme activity compared to only 103 enzymatic activity in the heat-treated infant formula. We know the breast milk contains large number of endogenous enzymes, so it will be another benefit for this enzyme, and also for other heat-liable ingredients like vitamins, especially water-soluble vitamins and lactoferrins. Other future works including optimizing our processing for the industry scale, and also validates the ex vivo in vitro cell culture model for bioassay, and also assess the stage 2 infant formula. How is the stage 2 infant formula with 50-50 whey protein and casein ratio shows also different digestibility and the growth rate. I would like to thank you, my supervisors, Andre, Mick, Linda, and John Tobin, and I show the picture of my examiners, Xiaodong Chen from Suzhou University, my hometown university, and Flora from MTU, and the chair Suzanne. Also, six team members give me a lot of help during all my trials.
Thank you, Yihong, for a very interesting talk. And you were discussing the use of membrane filtration as a way of improving protein digestibility of infant formulas. And you did a very interdisciplinary complex work where you used a simulated in vitro digestion model. You all went on to use absorption models, and you also had an in vivo setup with piglets. So that was quite good work there. We want to open up our Q&A session, and I would like to encourage us all to pop in our questions in our Q&A, and we will get to answer that. I'll just kick that off by asking you, Yihong, since you did all this interdisciplinary work, I would like to know, did you use the adult model for Infogest, or did you alter the parameters of your in vitro setup?
Thank you for your question. Yes, I do use I change the model to the infant formula model, like reduce reduce the activity of the enzyme and change the secretion male to secretion ratio, kind like so thing, based on several literature what we have for the moment, and also due to the infant has a higher faster gastric pH. So I also have to change their initial pH in their gastric model, and also the following pH declaration need to use other calculation, need to use other formula compared to the audience as well. Yeah.
Okay, interesting. Thank you so much for your answer. We do have one question in our Q&A. And Curata is asking, what do you think if the compounds in form of polyphenols encapsulated with liposome in gastric digestion, will it be the same like protein digestibility? So I think she's talking about polyphenols, which can be linked or encapsulated in liposomes. If it would be the same as you saw with your protein digestibility.
So if it's for the encapsulated polyphenol, I will think this part will more affect the free amino acid parts. Is I'm not sure. I'm not sure if it's correct. I think the member filtrated system maybe not affect the encapsulated material, because they are not they probably being felt out, or we say they are in the retentate phase in when we treat our infant formula with their member filtrated system. So these parts may be added afterwards, after the main prepared stream being member filtrated. So I'm not sure, but I don't think that part will be affected. But also the encapsule maybe perform like a fat droplets. It can also affect by the native and the natural whey protein. It's quite interesting question. We probably need to see.
Yeah, right. Thank you. And talking about membrane filtration, I think we saw from your slides that you were doing this in a pilot plant. Do you think it's now a commercially widely used application, the membrane filtration?
Yes,
in industry?
It does. I know, as I know, there's many company has using the member filtration system to produce skim milk, and the purpose of that is to preserve the native material in the milk. And the customer reply was they are these member filtrated skim milk has better flavor compared to traditional heat treated. And also, when I make my infant formula after we made the powder, the flavor smell much better compared to the heat treated infant formula as well. So so for the industry, for the skim milk, to making the skim milk, they definitely already using the member filtrated system, and there's many company also interested in to using the member filtration system to make their infant formula as well.
Yeah. Okay. Yes, that's good. And we have George with a question. He says, great work and a very interesting presentation, Yihong. And he highlights that in your presentation, you reason that there is a difference between casein emptying in both situations relating to their floating ability? He goes on to say that it is often mentioned that casein empties slower because of the large size of the casein flocks, which are retained by the antrum. Do you think that also plays a role?
It's what I think at here is it can play a role, but very difficult to play a role in infant formula because the infant formula only have about 1% 1% protein, and also the casein only contribute to 40% within this protein. So when the research to digest the milk, which contain about 4% protein with 80% casein, we do see some sedimentations and form the large gels, but when we digesting the infant formula, because the protein concentration is low, casein concentration is low as well. So I don't really think the casein. I don't think the casein aggregation will. The casein aggregation only can impact the digestion actually. Yeah.
Okay. Thanks, Yihong. So talking again with the differences between the membrane filtration and the heat-treated infant formula, do you think that the increases that you see respect to functionality and digestibility would have any sort of impact on the consumer and market perspective?
I beg your pardon for the question, please.
So I want to know whether the results that you obtained, that is the increase in functionality and digestion of proteins with membrane filtration, if that would have an impact on consumer and market perspectives.
Yes, well, yes, definitely. The research and from the customer feedback shows the infant formula increase their gastric reflex, so makes the infant uncomfortable compared to the breastfeed infant. So one of explanation give to this is because their infant formula had a lower gastric emptying. So because we have plenty of whey protein preserved in the native form, and which means a big part of infant formula can empty into the intestinal first. So I would presume in the future it could be one of benefits is it can reduce their gastric reflex. So it will be one benefit for the high native whey protein infant formula, and another benefits could be reduce the risk of their obesity or overweight because the high consumption of their energy. Yeah.
Yeah. All right. Thank you, Yihong. We have another question there, and they ask whether you just evaluated the bioavailability of free amino acids. Did you perhaps look to bioactive peptides?
Yes, we did experiments on the bioactive peptides test. So we extract 3 peptides from each each infant formula, and that three peptides is unique to the each infant formula, which cross over their bacillatory phase. So we presume they can from the in vitro results, we found that they can cross over their intestinal barrier and show up in the bacillatory phase. And we use a model we use an online model to estimate the bioactivity, and I pick 3 peptides has a higher potential bioactivity, and I tested that using the in vitro in vitro model, and the results showed these peptides, the 3 peptides overall from the member filtrated system, it can increase their adipogenesis activity for their adipocytes. Yeah.
Yeah. Okay. Yihong, you had quite very complex work. I would like to know what was the biggest challenge that you had in completing all of this, from the in vitro work to the absorption models to the in vivo protocol. What can you say to somebody who's starting out facing the same amount of complex work?
So the biggest challenge is the hypothesis I made, because the human body is so complex. It's a so complex interaction. So we do still need the magnificent study to find the relationship of the intestinal breaker to the brains, this hemostatic reaction. I don't have time to do that, but if I had time in the future, I definitely do this to make sure it does affect the processing, the digestion processing does affect the brain feedback to the intestinal and the following digestion, because this part couldn't show in vitro digestion and also if we're using the in vivo digestion, if it's difficult to observe as well from the piglets, because they couldn't give any feedback like a human. So it will be a very interesting point in the future.
All right, thank you. So you were looking at infant formula, Yihong, but the current trend now is more personalized nutrition, and a lot of people out there, a lot of research is being done to sort of like personalize nutrition to different groups. Do you think that this membrane filtration procedure would be applicable to other dairy products?
As I said, there's already skim milk. The skim milk product has been commercialized, and also I know some whey protein isolates products are also produced by the infant formula. So some research using the membrane filtration system to concentrate the casing, to try to use the filtrated casing to make the cheese as well. So definitely, the membrane filtration system become more and more common to using the dairy product industry.
Yeah. I'm not so sure if we have some more questions. We still have a little bit of time if we have some more questions. As we wait for maybe a few more if they are coming in. What's your next step in your career? It's always something, after you finish your PhD, it's always, oh, where do I go? So for you, what's the next step you're on?
I'm looking for a job in the infant formula area now. Yeah, definitely. I want to stick in this area.
All right. So you're really passionate about infant formulas and
yeah,
delivering
yes.
impact there. No, that's great, Yihong. Thank you so much. I I'm just noticing here that our questions listed in the Q&A are actually finished. So I will hand over to Talita.
Hi, everybody. I would like to thank all of you for attending our first Infogest Viva. Also, Yihong for presenting in our first webinar. We also wanted to thank Andre Brodikorb and Didier Dupont for the opportunity, and we are glad for being here to facilitate these webinars. We have 2 announcements. I'm going to share my screen. So The first one is about the special issue. We wanted to invite you to submit your paper to the special issue from the International Conference on Food Digestion that happened in May in Cork. The papers will be the accepted papers will be published in Food Research International, and all papers in on food digestion will be considered for this issue, even the ones who were not presented in the conference. We also wanted to announce our first Latin America Infogest trainee school that will happen in November in Chile, in the University of Chile. So it will be announced in the next 2 weeks on the LinkedIn and in the Infogest network, but it will be three days of school, and the ones interested, please send the application to this email here, and we will have limited number of places, so please submit. So we are going to select some of them to be there. Okay, so I'm going to stop share now, and I guess that's it. We wanted to thank you for being here, and we see all of you again in the next one in November. Thank you very much. Bye bye.